Thank you all. Hello. And just reminding that John Begg's homework can be found on the link and apparently on local computers as well. Wherever the local computers are, I have no idea. Okay? Maybe, is Victor there? No, Victor. No, he's supposed to know. I don't know. Okay? But you can download it from here. Apparently, he put it on this website. You can. It's a very long URL. I'll leave it there. Well, and like you just said, it's a very, I couldn't have chosen a better speaker before me like John Beggs. I mean, he just said everything. I don't have to give my talk, okay? I'll stay here, tell jokes for 2 hours. But actually, since he stole some of the figures I was going to show, that's okay. That's good. I can go back and revisit his daughter's picture on the refrigerator. And in fact, the thing you mentioned, just so you know that this is true, okay? I have, it was for a different talk, but since we have 2 hours, look, this is, look at this picture. Hold on. So this is a very strong magnet. And you take, this is another magnet, right? And you put it here, and you put it on a flame. It's very hot. So it's not ordered. There's no magnetization. And when it cools down, it goes below the critical temperature, and it's a magnet, right? So it jumps up, now again, in slow motion. And you can guess when it will reach the, oops, the other one is gone there. And then it cools down enough, the temperature is low enough so that the spins can align. You have a net magnetization, macroscopic magnetization, and then it will jump and be attracted to the other magnet. Right? So he's not telling just about a model. This is real, okay? It happens. And this is well studied. You see, John, you will burn your pictures, your daughter's picture here if you use this kind of flame for, you don't want to do that, okay? At home. Okay, so down to business. So you see that a significant fraction of my talk is to describe what neuronal avalanches are, which John has already done with much more competence than I could ever dream of doing. And so I can take my time here. So 2.5 weeks ago, I was in Mexico on another conference, and we have 1.5 hours to speak, and people were surprised to have so much time. Itamar Prakasha was telling this joke that 2 physicists, P1 and P2, were crossing the border of a country C, and they were arrested by the police with illegal drugs, right? And so and they were condemned to death immediately. So you're going to be executed. You have one last wish. What do you want? So physicist P1 says, you see, my whole life I was giving 15-minute talks, and nobody was paying attention because they have Wi-Fi and they have the laptops open. And my last wish, I want a 2-hour lecture with no Wi-Fi for anybody, and all the laptops will be closed. Okay? And the officer said, well, okay, that's fine. Physicist number 2, what do you want? I want to be executed first. Yeah? Yeah? So this is the scenario here, more or less, right? Okay. So a talk with such a title, The Rise and Fall of Directed Percolation, can only be divided in 2 parts. The rise and the fall, right? That's a natural thing to do. So what I'm trying to do here is revisit some of the things that John talked about, visit other things, and try to be more on the side of the models and try to see what kind of things have we tried, what is the success of models in reproducing the data, more or less like this, and give you some more data and a little model. So let us start with these neuronal avalanches in cortical slices. Let me stress this, anesthetized or resting animals. So cortical slices, anesthetized or resting animals. First, I like to start by acknowledging before I forget. So I'm going to talk about work from other people, and the work I have done here is mainly with Osamu Kinouchi, this guy here, our friend. And these are the people who paid for everything. And later on, I'll talk in the second part, in the fall part, I'll talk about work that I've done with Siddhartha and Thiago Ribeiro here. I'll come back to that. So, neuronal avalanches in cortical slices anesthetized or resting animals. John has told you most of those things. I'll just revisit them and talk a little about them. So it all started with their paper in 2003, Journal of Neuroscience. You have 60 electrodes here in a cortical slice. So this is in vitro behavior, right? It's a petri dish. And this is one of their pictures. Here is time. Here are the 60 electrodes. And you see some bursts of activity here. And then some silence. And then some and it looks synchronous, right? It looks like everything is at the same time. Everybody together. It turns out it's not really synchronous. If you look at a finer temporal scale, you see that some electrodes light up, some others don't light up. And what I forgot to stress in the previous slide, I'm sorry, these are local field potential. So like John mentioned, a local field potential is the average, so some kind of average, electrical activity around an electrode. So it samples from hundreds to thousands of neurons around it, some kind of average electrical potential. And so when you take this data and you bin with an appropriate bin, what is appropriate bin? Well, for instance, what they have done in this paper is that you can, for instance, take the average interspike interval here as a reasonable bin to bin your data, because then you will be neither too large a bin. If you take a bin this size, it's never silence, right? If you take a bin like 1 femtosecond, it's always silence. So there are no avalanches. So in which sense this is silence, and this is non-silence, as long as you define some temporal bin. I'll come back to that later. So if you define a reasonable bin from the data, you get something like this. So this is what they call an avalanche. So you have silence before and silence after, and some activity in between. And from the picture here, you can accept pretty quickly that so these neuronal avalanches have this separation of time scales, in short, STS. So what that means is that the duration of the avalanches is much shorter than the recurrence interval. This is one of the features that it shares with real avalanches. So it's like, I don't know, snow avalanches, we don't have that many in Brazil, but snow has to accumulate for, I don't know, months, weeks, and then an avalanche takes some minutes or seconds, and then it's over, right? So it's much faster than the accumulation process. And this emerges naturally in this system. Okay, so, oops. So one question that you could ask, a natural question, so I have 6 electrodes. So on average, how many electrodes are active in a given avalanche? That's an obvious question to ask, right? So what is the typical size of an avalanche? How many? And so that's John has described already. So you define the size of the avalanche as the number of electrodes that somehow lit up here, and you do the statistics. And when you do the statistics, what you obtain is that the distribution, this is probability of an avalanche having a size S, is a power law, or very well described by a power law with a cutoff here. And the cutoff changes with the number of electrodes that you choose. And if you define an avalanche, the size of an avalanche in a different way, like summing the amplitudes of the LFP deviations, it also gives you the same thing. And you can play with the system like you would in a simulation, except that this is real data, and the cutoff changes the way you would expect, more or less. It's very nice. So, given that the distribution avalanche size is a power law, with an exponent 3/2, you can go back and revisit that obvious question that you were asking before. So, typically, what is the size of an avalanche? A typical size. Come on, I have 6 electrodes. Typically. The answer is that, in light of this distribution, is that, well, this is what we call a fat tail distribution. It has a fat tail, right? It doesn't decay exponentially. It's much slower than exponential. So questions like the mean size of an avalanche, the typical size, the mean plus or minus standard deviation, they're just meaningless questions. Because for such a distribution, you can do the calculations. If you try to calculate the standard deviation, for instance, it's infinite. So if the standard deviation is infinite, what sense does it make to ask what is the mean size? Well, I can give you the mean size, but it doesn't mean anything, right? And I'll try to revisit and complement John's comments about why should I care. Well, you should care because if the mean doesn't mean anything, you shouldn't calculate the mean. You cannot base your stuff on, okay, well, what is the mean behavior? Well, the mean behavior doesn't mean anything. This system is governed by fluctuations. Fluctuations are very large. Fluctuations dominate the system. And why is this relevant? Well, because if you're seeing this in a piece of brain, maybe that's how the brain operates. Maybe, we don't know. But maybe that's how the brain operates. And that should tell you something about how the brain does not behave, for instance, right? So it doesn't work in chunks of identical packages, for instance, apparently. So that's important. In the way in our attempts to understand neural code, whatever neural code is, or whether it exists at all. So we don't know, right? So you can go fancy with words and say that this is a non-trivial scale invariant statistics. Essentially, that means it's a non-trivial distribution. If you take a bunch of neighbors and connect them in a simple way, as John has shown, you will not get this. You get an exponential distribution. So avalanches would have a typical size, but they don't. Right? Okay. And so these results were very nice. They caused a huge impact in the field because, let me show you something else. So there's another way to characterize this kind of activity. Well, Jesus, I'm sorry. This kind of activity, which is not exactly a wave, it's not exactly synchronous, it's something else. It's an avalanche. So one of the parameters you can use to characterize this is the branching ratio. So essentially, once you have binned the data, you see how many electrodes are active at frame t, and then at t plus delta t, the next frame. So this is the number of ancestors electrodes, active electrodes, or spikes, if I talk about spikes, not the case here. And this is the number of descendant spikes. So you have something at this time, and then they branch somehow, this activity propagates, and you have some other activity here. If you divide this by that, you have the branching ratio. And experimentally, it fluctuates around 1. There's a huge disclaimer here that estimating this thing experimentally is a nightmare. It can give you all kinds of trouble. And it changes a lot if you change the time bin. But for this time bin, it's very nice. It fluctuates around 1. 1, meaning if you interpret this correctly, is that, so on average, I have like 5 spikes now, say, and the next time step, I'll have on average 5. So it's not increasing nor decreasing. It's kind of in the balanced. Say, if this was smaller than 1, if for every 10 spikes I get 9 next time step, guess what happens? In a few time steps, it's over, right? Everybody dies out. This is what we're going to call subcritical. If sigma were larger than 1, say 1.5, I have 10 now. Next time step, I have 15. And then I have 15 times 1.5, which I don't know how much it is. And then, it will explode. This will be what we call supercritical. And this is akin to, say, a nuclear reaction in a nuclear bomb. That's how it works. So energy from the neutrons, from nuclei, excite other nuclei, etc. And this goes back a little bit to your question about self-organized critical models. Because in those models, now we know 20 years later, after the 90s, that they have to have sigma equals 1, which in the models translates into the model, in physics translates into the model being conservative. Otherwise, they're not critical. The forest fire model, for instance, has trouble with that, right? The forest fire model has trouble with that. So, yeah, there are several constraints from theory that you get. Even though now we are revisiting that. It's a different paper. I'm not talking about it, but we can talk later maybe. Yeah? No. Well. The slice is two-dimensional, yes, but the connectivity we don't know. Right? The connectivity we don't know. It certainly is, it has small world features. I can bet another beer, right, John? Yeah. It's probably a small world because it's not local and you have long-range connections. But other than that, I don't know. Why are you asking about the dimensions? Yeah. Yeah, but that's okay. But the branching ratio is more or less, the definition is dependent of the dimensionality, right? So it just measures the outcome of whatever is going on in the underlying topology, so which we don't know. Okay? And like I said, it's a very problematic measurement, right? So Viola Prisman published a paper showing that this can be troublesome. And so this was in a slice in vitro. You can do it in vivo. If you take anesthetized rats and measure local field potentials, you also get a power law. Again, always with this caveat that was mentioned before, that you have a few order of magnitude because that's the experimental constraint of the number of electrodes, essentially, that you've got. And then there was this other paper, I think this is from Nicolelli's lab, where they measure the premotor cortex of monkeys, which are very well-trained monkeys, so they can stay sitting down for hours, or like just drinking juice, and they'll be quiet. My children cannot do that, for instance, right? And so it's very important to say that those monkeys are anesthetized, so they're awake monkeys, but they are resting. So you're measuring brain regions which are involved with movements, with motor control, and they're not moving, so they're just quiet. And you see power law distributions like this. Well, things that, if you don't like me calling them power laws, we can say that, well, this is non-trivial statistics because it's not exponential. This is exponential. This is what you get if you shuffle the data. Okay? So you get something there. And then this has an appeal to physicists because in the 90s, people were interested in all those power laws. For instance, the forest fires. This is not a model. This is experimental results from Malamud and all, a very famous paper in Science. You see this power law distributions. And the sand piles that Perbach proposed didn't work, so they didn't give a power law, but rice piles apparently do. don't ask me why. So, well, to some extent here. So you have these, some kinds of rice, actually. And you see this power law. So there was interest, because then in the 90s, there was huge literature on self-organized criticality and how those power laws would emerge. And so, in a way, the paper by John Beggs and Dietmar Plans kind of gave a new life to this field, which could then connect those ideas of self-organized criticality with the brain. And also solar flares. So, okay, solar flares are also power law distributed. Solar Physics, 1985 paper. So I'll try to revisit what John said and try to revisit what I like to call this critical brain hypothesis or conjecture, if you want. And to say why does it matter, and then try to discuss some of what Don Katz was asking us about yesterday, about this modularity thing, which is a problem, and see how this sheds a little light on it, maybe, just maybe. So, roughly speaking, this critical brain hypothesis is, like John said, is the idea that the brain as a dynamical system operates near a critical point. So this is the brain, this is the critical point. If you want, this idea can go back to Turing if you stretch it a little bit, but Turing said something about the way people talk and converse and how people produce ideas, and he talks about supercritical people, essentially, in the way they produce ideas. And then there was Perbach and Dante Kelvin in the 90s, and these two papers here, Nature Physics 2010 and Xu and Play's Neuroscience 2013, are two recent review papers which go back to this history and try to tell you everything in detail. So if you want to know more about this field, those two papers are review papers. And so the idea here is that, well, John has shown you some, I'm not sure he has shown this bifurcation diagram. No. So the idea is that you have here some control parameter. For instance, in his example, the temperature. This will be a control parameter. You can change the temperature here. And you observe what happens to what we call in physics, an order parameter, something which characterizes the order in your system. In the case of his example, the magnetization of a magnet, right? So this will be 1 over temperature, okay? This is low temperature, this is high temperature. For low temperature, you have magnetization. For high temperature, you don't have magnetization. And it's not continuous, well, it's not asymptotically. You have a critical point where things change qualitatively. And in this particular point, as he was showing you, you have some very special features, right? Strange things happen here, very interesting things happening here that gave jobs to many physicists in physics departments over the last decades. We were studying this point here. So this is the picture of a second-order phase transition. And this red point is what we call a critical point. And we call second-order phase transition when this transition here is continuous. It could be discontinuous like this, and it would be a first-order phase transition. Okay, so this is just hand-waving, blah, 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 right? Because in the brain, what is the control parameter? Well, parameters, probably, right? So many parameters in the brain. What is the order parameter, or are the order parameters? We don't know. We don't know. Really, we don't know. We can try to guess. And we've been trying. I mean, the community has been trying for... John's paper is now 10 years old, 11 years old. So that's what I'm trying to revisit. So if you try to do theory, and the brain desperately needs a theory, I think this is why we are all here, I think, in a way. People have been trying to estimate those things, or come up with ideas. And the title of my talk is Rise and Death of Directed Percolation, because it is one of the class of models that people have been using to describe what's going on here. And it's been very successful up to a point. And I want to show you the fall of the model a little bit, because it's time you come up with new models, essentially, I think. My opinion, at least. So, phase transition 101, crash course, one slide on phase transitions. Fortunately, John has done a much better job previously. So the idea is that if you are at a critical point here, or near a critical point, you have those non-trivial scale invariant behavior, which you can characterize, among other things, by critical exponents. So for instance, if I'm giving names now, okay, I'll call F0 this order parameter. In the vicinity of this critical point, the way it departs from, as a function of sigma, is sigma minus sigma critical, which is this value here, to the power beta, where beta is a critical exponent. And if, for instance, if you are right at a critical point, and now let's put a third axis here, which is the external stimulus. So I'm stimulating this network right now, okay? The way this activity will go up, right here, is nonlinear. It's the activity, the intensity of your stimulus to some power 1 over delta H. And this is another critical exponent. John was mentioning the correlation length. Essentially what happens here is that correlation as a function of the distance x decays exponentially with x, with some correlation length psi. Now, psi as a function of the distance to this critical point, psi diverges as you approach the critical point, and it diverges with another exponent, which is the divergent correlation, so the new perpendicular, it's called in this literature. And if you correlation times also diverge. And another exponent, so there are several exponents. I'm not listing them all, so I don't kill you with boredom, but there are several exponents. And one of them is the exponent which governs the size distribution of avalanches. If you take your system and you perturb, this perturbation creates an avalanche, you calculate size distribution, you have an exponent. Okay? That's what you expect if you are here. And this is the result that John has shown before. I've just shown you again. So this is physics. Now, why would you expect the hypothesis to be true, namely that the brain should be here? Why does it make any sense? John has told you the story before. I'm going to repeat it again. So the theory predicts that scaling invariant dynamics here, so models, provide optimal transmission capacity. So John has shown in the very first paper, actually, John and Dietmar. Also, optimal information processing, like he has shown before, in very nice neurocomputational paper. They have shown in a physical review letters that you have the largest repertoire. So you can understand repertoire as for memory storage, for instance, some kind of memory, or for recruit, or as processing diversity for the information processing here, right? And then Woody Shu in Dietmar Plan's lab has also shown that this is true experimentally in the slices. And what we showed with Osamu Kinouchi is that you also have optimal sensitivity and dynamic range to incoming stimuli. So this is the part where you're no longer necessarily talking about a slice that doesn't do anything, but as an operating brain, which I think is interesting. So if you just look at these features, optimal transmission capacity, information processing, repertoire, of dynamical repertoire, sensitivity and dynamic range to stimuli, well, those look nice, right? If I had a brain, I would like to have those features, right? It would be nice if my brain had those features. It would be a good brain, apparently. So the reasoning goes that if we accept that these are nice, these are all highly desirable for a brain, therefore, there will be evolutionary pressure, and Saint Darwin will take us there. Somehow we would evolve, and we'll come up with mechanisms that will take you to this point, which again goes back to one of your questions, how you get there. So besides the Hermann-Levina-Geisel model, there are several other models just trying to show that, yeah, you can be close to this point. And actually, for biology, it doesn't really matter if it's really critical or if you just fluctuate around this point. I'm not sure it matters, really. But there are mechanisms that suggest that you're here. Right. So, okay, so this is very blah, 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 hand waving, will be very nice. So do we have a concrete model? So yes, we have several. And the historical model, I mean, if you revisit John's original paper, etc., is one which I loosely call the directed percolation models, class of models. Why? why do I say class of models? Because when you, I didn't mention before, I'm sorry, these critical exponents here are very weakly dependent on the details of your model. So you come up with your model, and it has a phase transition. You calculate the critical exponents. Okay, it gives you something. Now, I want to publish another very quick paper. So what do? I take her model, and I modify just a little bit, right? Put some extra ingredients, just some, it doesn't change really much. Well, it changed the critical parameter stuff, but it probably will not change the critical exponents, which will be exactly the same. So they are independent of details. They are dependent on very few things. Like dimensionality, has to do with Gilberto's question, has to do with symmetry of what is involved, symmetry of your dynamics and stuff like this. So when two different systems have a phase transition and have the same exponents, they're said to be in the same universality class. Okay? And then some more concepts on statistical physics. I don't know how much you guys know about this, but since they are biologists, we could revisit those points. So I'm talking about one universality class, one class of models here. So, which is the model I've used for a long time. So this is a cartoon of what happens when a neuron spikes, right? The neuron is quiet, doing nothing, it has resting potential, and then something happens, some current comes in through a synapse, and if it's above a certain threshold, it can spike, and then it goes refractory for some period, and then eventually comes back to rest. So there are, if you want, you have Hodgkin-Huck equations, you have complicated equations to describe this thing, but since I want to model a large number of neurons, I cannot afford to simulate 10,000 of those Hodgkin-Huck equations, and I also have the faith from statistical physics that maybe I don't need to, because if I throw away everything and just keep the essentials, maybe I'll get the same results. So that's what decades of statistical physics also teaches, that you can throw away several things. There's a leap of faith, a little bit, but that's what you do. You do take the leap, and then you come back and check eventually. Suppose you're spending, I don't know, how gazillions of euros in building a very perfect simulation of the brain. Suppose it's infinitely perfect. So you finish simulation, it will talk to you, right? Hi, I'm the simulation, right? And what have you learned about the brain exactly? I don't know, right? That brains can speak, but we knew that already. So this thing off? It's off, right? That's, oh, okay. So the mimic here is a cellular automaton model. So quiescence is 0, spiking is 1, and refractory period is 2, 2, 3, 4 to the minus 1, n minus 1, and then it comes back here. And it only jumps from here to here if it is simulated, and the rest is pretty much deterministic, right? So this is one neuron. One very dumb neuron, very stupid neuron. Now you take those, one of these here, and put on a lattice, right? Some graph. I'll talk about random graph because it's just the easy thing that you can do, perhaps. This model is heavily based on 2005 paper by John, is it Klaus, his first name? Klaus Haldeman? Haldeman and John Beggs. So the idea is that this is a neuron, this is a neuron, this is a neuron, this is a neuron. You have the cellular automaton on each side, and so if this guy has k equals 4, he has 4 neighbors, that's what it means, ki equals 4. So this is the probability pi1 connecting neuron I to neuron 1 and 0.1. What that means that if this guy spikes, and if this guy is quiescent, then it will spike in the next time step with probability 0.1, right? This will spike with probability 0.9, 0.2, 0.5, etc. Okay? And then from here, you can very naturally define the local branching ratio. Remember that I mentioned before? For neuron I, which is just the sum of these pi. So 0.1 plus 0.9 plus 0.2 plus 0.5 hopefully gives you 1.7. Yeah, that's right. So on average, if this guy spikes, it will generate on average 1.7 spikes, right? If you take an ensemble of the realizations here. Okay? So you put this in a random graph, so every site has some, on average, k neighbors. And bear with me, I hope this is, okay, there are no more equations after this, okay? I promise. So you can define a very natural control parameter here, which is the average branching ratio, so the average over all sites, which is sigma. And you can control define another parameter, which is the firing rate, average firing rate. So it's just the probability that a site is in state 1, which is spiking, essentially, right? And you can do physics, you can do calculations. These are the mean field equations I don't want, yeah? No, I don't compute branch ratio at all. I just take it from the connections. Yeah, it becomes a parameter of my model. I just define sigma, and then I do a calculation backwards, and then this defines the p's more or less, right? Yeah, so I have no guarantee that sigma that I put in as a parameter is the sigma that you would measure if you did. But then again, I'm not sure we should care, huge parentheses, because first, it's very hard to measure experimentally. Second, people that have shown that in a different topology, more complicated than that, say a scale-free network, for instance, sigma doesn't tell you anything. What tells you something is the largest eigenvalue of the connection matrix. So maybe, it's just, sigma is very useful for talks, because it's very easy to explain what it means. But in practice, I'm not sure it has the relevance people have attributed to it in current days of complex graphs. Right? So obviously, I'm not doing the calculations here, but you can do calculations. So this site here, besides being simulated by the neighbors, we can also put some external stimulus, which I come back to later, okay? You can simulate externally, and essentially, you have a self-consistent equation where you show that the probability that I'm firing now depends on the probability that other neurons are firing and how they transmit their firing to me, right? So it's self-consistent equation. You can solve that if you want. And I was going to put this in the exercises, but I didn't, okay? So I'm being nice. Actually, I forgot, but I have homework for you. And so would you please pass along to the students? Take one and pass along so you can take hints during the talk of what you have. It's just two very simple calculations. No calculus required, okay? No calculus required. Just a calculator and a ruler or your thumb, maybe. Okay, in the plots? Okay? Very simple. The text is long because I'm trying to be pedagogical and stuff, right? Okay, so my order parameter here is, I'm calling F0, the firing rate when I don't have external stimulus. So whatever the network is doing on its own, thanks to activity due to other neurons, okay? If you take this equation, so this, look, again, revisiting, what is this equation relating? It's relating the order parameter F, mean activity, mean firing rate, to the control parameter, which is the branching ratio, the intensity of the connections, right? Think of sigma as the intensity of the connections. You solve this equation, and you get this, right? This is the picture I've been showing you all along. So spontaneous activity of the network as a function of the coupling is 0 if the coupling is too weak, and it's non-zero if it's supercritical. So this is supercritical, this is supercritical, right? Essentially because of the scenario that John has already shown you, a subcritical system, so sigma less than 1, you create less than you receive, well, activity eventually dies out. If you create more than you receive, activity would explode if it were not for the refractory period. So refractory period prevents this from creating an explosion, and this thing saturates at some point here. And right in the middle, at a critical value, you have the critical case. Okay? So this model is very similar in spirit to the branching process, which is a model designed in 19th century. Who was born here? Okay. To explain the survival of family names in Britain, right? What? Yeah, Galton-Watson process, yes, the Galton-Watson process, which was then revisited and, well, reinvented, put it on a lattice by Harris in 74, and then this kind of phase transition nowadays is called, well, it belongs to the directed percolation universality class, in the sense that it doesn't matter how much you change your model, well, if you don't change too much, it will give you the same exponents as the directed percolation model, this kind of model. Another way to look at this phase transition is like this. So, I'm not sure you can see here. So this is rho, this is the density of active sites as a function of time. So if I choose sigma 0.95, this is subcritical, and I put like 10% of the neurons firing, right? This is a log scale. Well, activity dies out very quickly. You see? These are 3 different runs. You try again, you try again, you try again, it dies. Not very interesting. So activity propagates a little bit and then eventually it dies. Now, if sigma, the coupling, is slightly supercritical, slightly above 1, that 10% activity now stays in the network. Because I'm stimulating my friends are stimulating their friends with even more activity, right? So activity is being amplified all the time, so this thing never dies. Only because of your initial stimulus. So it stays there forever. And in the critical case, it dies. But you see that it takes a longer time to die, and the variation of the time is also large. So actually you can calculate the distributions of the survival times here, and it will also give you a power law. Okay? So the behavior here is non-trivial. So what we have to know about this universality class, directed percolation universality class, is that it's a phase transition very different from the one John was showing for the magnet. Very different. It's a different. Why? Because there you either have disorder, spins up, spins down, and then everybody like this. And here I have a transition between something which is, okay, you can call it disorder if you want. I call it order, right? Because here you have neurons spiking. And the other is not disorder. The other side of the transition is death. Your neurons are all quiet. It's very different from fluctuating spin models. So this is, in the literature, it's called an absorbing state. If you put all your neurons quiet and you don't do anything, that's it. End of the story. You can stop your simulation, because nothing else is going to happen. So it's a different class of model, different universality class. And another way to understand this is, again, remember I told you that universality classes rely on symmetries. So the Ising model has up-down symmetry. So spins are up and down. And if you take the system and flip it upside down, your magnet, it's still a magnet, right? So there's no real difference between up and down. Right? Here, dead or alive are not symmetric. Right? We know that, so spiking neurons and quiet neurons are not the same. One is not, one minus the other. It's just not multiplied by -1. So they lack this up-down symmetry, and this changes the universality class and changes the exponents. So, now, you take this model here, this model or any other model which looks like this would belong to the universality class of directed percolation. So you put in the critical point. And then you introduce separation of time scales. Remember what it is? So an avalanche, the interval between avalanches is much longer than the duration of the avalanche. How do you do this? Well, I do like this. Well, my system is quiescent, my simulation, right? It's a model. So I create an avalanche, and then the avalanche is over. I wait until it's over. I'm looking, I'm simulating, it's my simulation. I wait until it's over, calculate the avalanche size, and then do it again, another avalanche. I create another one. So I impose this by hand. Okay? If I do this and calculate the avalanche size distribution, blah, 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 you know what's coming. You get a power law distribution. Right? This is work by Daniel Larimore, 2012, where they show you how to calculate this thing for the general case of a general topology instead of the simple Galton-Watson process. Now, experimental results, as John has shown before, I'm showing now, you also get the same power law with the same exponent 3/2. Okay? Except that note that now separation of time scales obviously emerges. They don't wait until the avalanche is over and then simulate again their in vitro slice. It emerges. It's there. It's intrinsic to the system. So, so this thing here, the idea that this thing looks like this, is what gave the main link between those two ideas. Namely, so neuronal avalanches, experimental ones, are compatible with critical dynamics. And the exponent is the same. So what John and Dietmar measured here is an exponent 3/2. So you can revisit the model and take, okay, what do we know about the model? What do we know about the exponents? Well, what we know about the exponents, it's very well studied for decades. It's a very old review from 1999 already, where they put together all the critical exponents together with exponents for the avalanches, which somehow had been lost. It is almost as though the self-organized criticality community didn't connect with the critical phenomena, and some people then did this job of theoretically connecting. Remember that John told you that the exponents are all connected? So in a given universality class, they are all connected, including tau. So they're not independent. And that's what he was showing. You have to show that there is some connection between them. And you see another digression in statistical physics, but that's what we do here. So if you take this model I show in one dimension, you can have a phase transition, and it will give you an exponent, an avalanche exponent of 1.1. If you do in two dimensions, it will give you 1.268. In 3 dimensions, 1.395. In 4 dimensions, it gives you 3/2. In 5 dimensions, it gives you 3/2. In 6 dimensions, it gives you 3/2. And 3/2 forever, because you've reached here what is called the upper critical dimension. The exponents don't change. The exponents don't change anymore once you reach this point. And very interestingly, that's the exponent you get experimentally. The exponent for d equals 4, and for any other complex topology, like a random graph, or like a small world random graph, a small world network, where you have these long-range connections. This is enough to mix up the dynamics so it doesn't look three-dimensional, two-dimensional, one-dimensional anymore. It looks like mean field, as we call them. Yeah. Yes. Like Barabasi-Albert law. Yeah, it is. Yeah, it is. And this paper I mentioned before, Daniel Larimore, they do this calculation and with some very weak assumptions on the underlying topology. It has to be well connected and irreducible, blah, blah, blah. But it's yeah. For instance, yeah, it gives you this exponent, right? So this is one of the reason I'm showing this table is that the fact that this d equals 4 is the upper critical dimension exponent means that the data is giving us relatively little information because several models could fall into this into the mean field universality class, so to speak. Yes. No, because if you could have not direct percolation, but this other percolation, dynamic percolation, which is a different universality class here, but here is the same. Well, so several different theories, 7 different universality class meet at the mean field. And then, since you have mean field exponent experimentally, you cannot, this does not differentiate between the models. You see? It doesn't give you predictive power of the model. Well, several of them can give you a mean field exponent. That's what I mean. Yes, yeah. Actually, John's results is interesting because he obtains something there which is not exactly mean field. So this sheds some light on the problem. Okay, still a little blah, 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 because, okay, let me try to give an example. So the reason why there's a critical brain hypothesis is the rationale is that, okay, if it were critical, it would have very nice features. So do brains have nice features that we can check and compare with critical systems? So let me show you an example. So this is first a theoretical critical brain and try to see if there's anything with reality. So if I revisit that slide on phase transitions, let me talk about this. The response to a stimulus at a critical point here. And let me run a very simple simulation where what I'm showing here is 50 by 50 neurons, like the cellular automaton neurons, very simple one. And this axis here, this is the same plot, okay? This is log, this is linear log. And the horizontal axis, I have what I call the stimulus rate. And in the vertical axis, I'm going to show the mean activity of this network. So I'm interested in the response function, right? So if neurons are not connected, this is very boring and uninteresting. So low stimulus means that you're stimulating with some very slow rate. So you stimulate somewhere, the neuron spikes and goes refractory, and stimulate some other place, the neuron spikes. One stimulus, one spike, boring. Okay? So I'll try a stronger stimulus. What do stronger stimulus mean? So this happens more often. So if I stimulate with the stronger stimulus, I have more spikes. Boring. Obviously, right? Yeah, I mean, yeah. So imagine this was your olfactory epithelium, right? Which is the example that we use in our paper, actually. So, and the things arriving are odorant molecules, and when it binds to your olfactory receptors, it will spike, right? So if this were your nose, this will be the responsive function of your olfactory neurons, and this will be the odorant concentration, for instance, right? It will be, you see, it's a linear function, because I stimulate here, it spikes a little bit. I stimulate like twice as much, I get twice as many spikes. Boring. Actually, that's what you see in olfactory neurons of the frog, for instance. If you really kick in a very strong stimulus, what you get is saturation, because now you have a refractory period which prevents your neuron from spiking too fast, right? But it's still pretty boring. So I'll try to convince you that this cannot be your nose. It cannot be like this. So for now, just a model. So now I'm going to couple those neurons, and I just made a square lattice like this, so it's easier to show, right? So I'm going to couple those neurons with their neighbors with some probability, some small value of sigma, okay? What happens now? You see, now I stimulate. Whenever a stimulus arrives here, because of the connections, now this creates a little small wave, right, which propagates a little bit. So, and then it dies because it's not strong enough, this coupling. So what happens here, from the point of view of sensory processing, is that if this were your nose, now, because of the coupling, instead of for each stimulus I get one spike, for each stimulus I get a bunch of spikes now. Right? Because of this amplification. So that means that the signal that your brain is receiving, which is this, not this, is a little bit amplified with respect to what it was before, because you're amplifying. So weak stimulus is amplified, strong stimulus is not amplified because you're saturating anyway. So this looks nice, right? So let's amplify even more. So if I take stronger stimulus, now I create larger waves, right? You see, because there's more probability that this activity will continue for some time. And then what you get as a response is, well, even larger amplification of the stimulus here. Okay? But still, boring because it's still linear in the sense that previously I had like one stimulus, one spike, two stimulus, two spikes. Now I have one stimulus, 4.2 spikes, two stimulus, 8.4 spikes, right? Did the math right? Yeah, okay. Advanced math, right? Okay? So it's amplified, but it's still linear. But you see, what am I doing here? I keep increasing the connectivity, the intensity of the connection, and these waves, they get larger, right? They get larger, they get larger. So what happens if I keep increasing the connection? Well, we know the answer because I've shown it before, but so if I keep increasing the connection, I'll reach a point where I reach the critical coupling. And then to show you here, I'm simulating a very, very, very low stimulus, which I know is so weak it will only generate the first spike here at 17 milliseconds, I think. Oops. Oops. Oh, I have to do it again, right? Okay, forget it. Now, yeah. See? Now, it creates one spike here, one stimulus. And you see, this thing is, another one here. It's propagating, and we don't know, is that wave going to die or not? Well, precisely, it is at the critical point. So it's on the verge of dying and not dying. At the critical point. And you expect strange things in this point. In particular, the response function here is no longer linear. You see, now this is a log-log plot, right? It's a log-log plot. So the response exponent is exponent. It's 1, 1, 1, 1. Oops, this is not 1. This is not 1 at this particular point. We'll see how much that is. If I keep increasing the coupling, what happens is that now I'm supercritical. And what this means is that if I stimulate with a very weak stimulus at 17 milliseconds, now I have so much coupling that this thing will generate this wave which will go on forever. So this is self-sustained activity, which can take several forms. For instance, it can be a spiral wave if it's in 2D, and if it's, this model can give you spiral waves. It can give you several. It can give you oscillatory collective behavior if you increase it a little bit, much more than that, right? But the point is that now, for any very weak stimulus that I put here, this thing goes berserk. Self-sustained activity is stable. If this was my nose, and at 17 milliseconds, this was one molecule of vinegar that flew from a bottle at the end of the room and hit my nose, that means I would be smelling vinegar for the rest of my life. And this is not a good nose, right? It's not a good nose. So let's revisit these curves here. When you put the simulation not on a square lattice but on a random graph, this is the same plots again, right? And I'm showing here. The symbols are simulations, the lines are calculations. For the random graph, you can do the calculation. It's very nice. You see that at this point, the response is 1/2. The exponent response is 1/2. And in the theory, it is a critical exponent. It's 1/delta H, right? What that means is that your response to a stimulus goes with the square root of the intensity of the stimulus. And you can try that at home if you want. Go to a dark room, light a candle. Don't look at the candle. Look at the wall and light a candle. And look how bright it looks to you. Now light a second candle. Don't look at the candle. Look at the wall. So you have twice as much light. And if you're honest, if you look at the wall, it doesn't look twice as much. It's less than twice as much. 2 candles look less than twice as much clear, right? It turns out that this response, the way we perceive an intensity of a stimulus as a function of a stimulus, is the oldest question posed by psychophysics in the 19th century, right? And they do the experiments exactly like I'm saying. So put it in a dark room, throw some light and measure how you respond to the intensity of the light. What's your perception of the intensity of the light versus the intensity of the light. And it gives you a power law as well. I can show you the results here. Again, I'm sorry. This 1/2 exponent, it's also a mean field exponent. And these are experimental results from a book on psychophysics. So there's a magnitude estimation of the brightness of a light as a function of the luminance in decibels. So this is a log-log scale. It doesn't look like one, but it is. And you can have exponents which are close to 1/2. Yeah? Account for what? Yeah, because Weber's law is stating that this is a log function, not a power law, right? So Weber's law essentially is the limit when this exponent goes to 0. And it turns out, well, this is a whole mess in psychophysics, okay? I don't want to take sides on this fight, but apparently, if you take a power law, which will eventually saturate, depending on where you fit your data, it can look like a logarithmic response. So I don't get Weber's law, I get Stevens' law from this model. Okay, but again, okay, so my model only works. Suppose I want to explain this, which is what we want in our paper. I want to explain this exponent. Where does the psychophysical exponent come from? Well, my model offers a solution. So if you put it here, in the critical point, you have the right exponent. So in physics, that's considered a very bad model, because of all the infinite values of sigma that I can choose, only one explains the data, right? Only if the model, at this point, you have the data explained. So why would that be the case? Let me talk about some other data which is not psychophysical. Psychophysics is very distant, right, from sensory neurons. So let's go to the retina. This is work in 2002 by Deans et al. And David Pohl. So what I'm showing here is different sections of the mouse retina. And what did I do here? Okay. Oops. So the idea is the following. The black here is because they injected a dye in a cell. And since those cells have gap junctions, so gap junctions is the physicist's dream of a synapse, because they obey third Newton's law, and it's just a connection between neurons, okay? And some channels connecting them, and ionic current flows from one to the other through a resistance. This is a gap junction, an electric synapse. And so you put dye in one cell, and then it diffuses through the channel and goes to another cell. This is just to show that there are gap junctions here. You put dye in only one neuron, and then it propagates to the other. And then it's a wild-type mouse. If you go to the knockout mouse, so it's you modify this mouse genetically so that it does not express the gene for connexin 36, which is the protein that makes those channels. So this mouse does not have gap junctions. You put dye in one cell and it stays in one cell, right? It doesn't go to the other ones. Just to show that the knockout worked, right? And now you simulate this retina. This is for those who don't know, the retina, you have several layers, and the last layer in processing is the ganglion cell layers, which then spike and send the signal to your brain, right, to your thalamus and then to the visual cortex, okay? So the last processing stage. The first processing stage is in the receptors, rods, cones. And what you see here is the response function, like I showed before, as a function of the stimulus. Isomerized molecules of rhodopsin per rods per second, also known as light, okay? This is light. And this is the normalized response function of the retinal cells. And you see that the wild-type cells have, for instance, this curve here, which is in your homework. You can have a look at it. I want you to have a look at this curve here. You see that, for instance, for a stimulus light, say, equals 10, it can already spike, right? It can already perceive something. You see some activity here. Whereas for the mouse, which lacks those lateral connections, at stimulus equals 10, you don't see anything. If I think about my model, I say, yeah, because here you don't have these waves amplifying what you're receiving, whereas here you do. One possible mechanism, gap junctions, right? And in fact, then I asked my undergraduate students to collect this data here, point by point, because the authors would not send us the results, so we had to extract from the plot. And instead of making it in a linear log plot, we replot this data here in a log-log plot. And it's here. Okay, it's just for 1 decade maybe, but with some good will, it has an exponent of 0.6, which is close to 0.5 that you predict with theory. Okay? So, so the model could have something to tell about the way you process information and why it is good to be critical. If you're critical, you have some non-trivial exponent here. A very more direct indication, but still nonetheless interesting, is work by Kihara et al. So essentially, this is a cut of the retina, and this green that you see here is the fluorescence for this protein which codes for gap junctions. This is the basal result that you see in the chick retina. Now, you do the following experiment. You put the chicken in the dark for 24 hours, right? And then you redo the experiment. What do you do if you were the chicken? If you're in this room for 24 hours in the dark, you want to see, right? You want to increase your sensitivity. You want to see better. And what did the chicken do? Well, not voluntarily, of course, but they express more gap junctions. So they increase the connectivity in order to increase the amplification of the signal. So increase your sensitivity. That makes sense? To my model, at least, it makes sense. It doesn't tell you it's critical, but it makes sense to me. Now, so why we should be here? Why we should be critical? I'm showing you evidence that, yeah, it's compatible with a nonlinear response. But why should that be the case? So John told you about this dynamic range. I'm going to revisit the concept. I have this response and stimulus, and it has some non-zero value, and then goes up and then saturates here. See, if this is light, and this is my response of my retinal ganglion cells, if I show you 10 to the minus 5 light and 10 to the minus 4 light, you cannot tell the difference because they give you the same response. Or if I show you this or this, they give you the same response, right? So these plateaus are trouble because they don't code very well. The response doesn't code what's coming. So a very crude way, you can be very sophisticated. You can do information theory, right, if you want. But if you don't want to do information theory, you just, okay, I'm going to discard things which are close to these plateaus. I don't like plateaus. So I just discard 10% here and 10% here. Whatever is left is good. So I can invert the function, so to speak. If I know this, I can know that. And this is the dynamic range, which is what I'm asking you to calculate with a ruler and a calculator from the plot. You take 10% above this value, come here and see, oh, this is the value. You take 10% below this value, oh, this is the value of the stimulus. So you see that 1 decade is a log axis, right? 1 decade, 2 decades, 2.2 decades. This is 22 decibels. That's the dynamic range for this toy curve. Now, come back to the model. Let's do it again with our fingers. So this is a family of curves, response curves of the model in which I'm changing the coupling this way, right? The coupling is coming like this. So no coupling, I cannot see weak stimulus. I increase the coupling, weak stimulus is amplified, and I can see it a little bit, right? If I couple too much, that's a phase transition. Now it goes up like this, right? And now it's a mess because now I have a plateau which I have to discard. So if I were to calculate the dynamic range from here to here, it's small, and then I increase the stimulus, the dynamic range increases, 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 and then it goes supercritical. And then I have a plateau. And then it's bad. And then it decreases. And then increase the coupling, the plateau goes up, I have to discount, and the dynamic range decreases, decreases, decreases. So dynamic range goes up here and goes down here. So the dynamic range as a function of the coupling has a peak at a critical value, right? And it's quite simple. So suppose I want you to hear this step here in the microphone. You can hear more or less, right? Can you hear? I'm moving very, very softly. And then I keep approaching the loudspeaker, which is there. And then eventually the loudspeaker will feed the microphone back, and hopefully you can hear a little bit better because there's some reverberation, right? This is the coupling. This is the coupling. So if I keep increasing the coupling by proximity, you'll keep hearing better, probably, if I could do the experiment correctly, right? Because there's some reverberation, which is the coupling, which is amplifying what I'm doing here, right? Now, if I get too close, and I promise I won't do that, if I get too close, what happens? Audio feedback, right? And what is audio feedback? Is the sound entering here, coming out here, entering here? So it's a self-sustained activity, which is beyond, regardless of what I do, right? And if I have audio feedback, I promise I won't do that. And I keep tapping the microphone. You cannot hear anything on what I'm tapping, because all you hear is audio feedback, which is what happens here in the supercritical coupling, right? Okay? So dynamics range is maximum in the model. Then, John already told the story. In 2006, Mar del Plata, Guiches City, there was a conference. I met Woody Shu in Dietmar Plan's lab, and I said, please, 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 could you please do the experiment? It would be so nice. Just take this slice and stimulate it. And 3 years later, very, very fast. So they did it. So the yellow lines here mean a stimulus in that slice, and the numbers here mean the intensity of the stimulus, just a current, a pulse of current. And you give a stimulus, have some response. You give a stimulus, have some response. You give a stimulus, have some response, right? A very strong stimulus, 150 microampere, and you have a very strong response. And then you can play with the network. Okay, so put some AP5 DNQX, which is glutamate antagonist, is that right? Yeah, okay. So this attenuates excitation, so my network is more subcritical-like. And then I stimulate, I have some weaker responses. Whereas in this case, picotoxin, I'm blocking inhibition, so the network is more excitable. So for the same stimulus, I get huge responses. Sometimes very strange responses, like this one, which they don't comment, and I do sometimes, like oscillating response, which I don't have in my model, remember? That's not the phase transition I got in my model. Okay, so you can calculate the dynamic range of these response curves, and you get like this. So I was happy for about a year, okay? Maybe more. I'm happy to today. Whenever I see this picture, I get very, very happy that it works. So most networks, no drug conditions, are around here. Kappa is more or less sigma. I didn't go into detail. Okay? Great. So, nice. So this is the rise, okay, of the model. Very nice. So everything, yeah, now the rise, like the dynamic range. Now the fall. So, seems to be working, right? So we have the model. That's the model. The control parameter is the Bernoulli ratio. The other parameter is the what is it? The other parameter? Yeah, the mean firing rate. So, so we went on to look at fingerprints of brain criticality in freely behaving animals. This is data from Siddhartha, not here. And the guy who did everything is Thiago, my foreign student. He's now at the NIH with the Dietmar plans. And also in collaboration with Fabio Cacheta, Indriel Belchior, and Dante Chialvo. And so there are main differences. So here we're talking about freely behaving rats. They can do whatever they want. They are awake, they can go to sleep across the sleep-wake cycle. Wake, slow-wave sleep, REM sleep. 3 different brain regions because Siddhartha, it's a different experiment for different issues. So we had V1 implanted, S1 implanted at the hippocampus. And the rats were in a box for some time. They knew the box, so the box had no novelty. You put some novel objects for 20 minutes, and then they go to sleep again. Possibly Siddhartha has shown you this picture before. And we also did anesthetize animals. I'll come back to later. And we did spikes, not local field potentials. So spikes, you only measure what's around your electrode. You don't cover your whole tissue. right? So this is what a raster looks like. You see, you could argue that you have separation of time scale here, maybe. So this is much shorter than that. But here, clearly, you don't. In a this is anesthesia. This is freely behaving animal. V1, S1. Clearly, you don't have avalanches in the rigorous term. What do you do if you're desperate? You just do it. You just operationally define an avalanche as whatever happens in between silences when you define an appropriate time being defined by the data. That's what we did, okay? Just close your eyes and go ahead and see what happens. So once you parse your data like this, you do have formally defined avalanches, even though they don't satisfy this criterion of separation of time scales. And you see, so this is an avalanche of size 2, avalanche of size 1, size 2, size 1, size 4, etc. And if you do the avalanche size as a function of time, as awake time, and look at only awake animals here, you see that there's a log scale. So it varies 2 orders of magnitude. So that gives you hope. Say, oh, okay. So yeah, so avalanche sizes are varying a lot. So let's cross our fingers and hope it's a power law, we get famous. Right? Or, yeah, it's critical. And then you do the statistics. You do the statistics? Yeah, okay. So in the 90s, like John said, you see you put a log plot here, you put the dots, and it's clearly a straight line, right? So that's what happened when I grew up. When I was doing my master with Nestor in the 90s, I went to physics conferences and people just pass a straight line here. It's not a straight line. It's not a power law, right? Actually, the best function we could find was log normals. What's very remarkable here is the following. I didn't have time to go in detail, but hippocampus S1 and V1 and pre-exposure, during the exposure, and post-exposure to novel objects, and whether the rat is awake, asleep, or REM, especially between those 3 colors here, these are very different behavioral states, right? That's the difference between an awake student and a sleeping student in your class. You know the difference, right? For the rat, it's the same, and electrophysiologically is very different. But yet, when you renormalize the bin, they look very much the same. The distribution looks so much the same that we were scared. Okay, so this is just bullshit. This is just an artifact. But actually, if you do shuffle your data, they get exponential. They get much smaller tails. So, okay, so what can you conclude about this thing, about the criticality in freely behaving animals? Nothing. Nothing. So what we did is, okay, let's make an effort to try to model this thing. Is there a way of saving criticality? Yeah. So what you do is the following. You take a model like I showed before. These are the dots, artificial, just cellular automata, right? They're connected. I make it critical because I control the model. It is critical, okay? And I have avalanche, and it's a power law. P of S is a function of S. It's a power law. Beautiful. It's my model, I can do whatever I want. Now, the blue data is the experimental data. Now, the experimental data is measured from electrodes. In this case, we had 16 electrodes. 1, 2, 3, 4, 1, 2, 3, 4, 16 electrodes. And each electrode samples just a bunch of neurons around those electrodes, right? And only, say, those red triangles here mean the neurons that you would sample in the model if the model was the brain and you had to measure with electrodes. So the electrodes don't measure everything. There's a huge undersampling or subsampling of the system. So if you only measure the avalanches in the red triangles in the model, which is critical, because I know it is critical, the size distribution that I get is the red curve here, which obviously fits the experimental data, otherwise I wouldn't be showing you, right? So that doesn't say anything about criticality, but it kind of, okay, maybe that's an undersampling problem. Okay. And then you can do other things like, so since this doesn't prove anything, can you do something else? Yes, you can, and it's an attempt to overcome the few orders of magnitude problem that you have because you have a small system, which is going to the time domain. Well, time, you have less limitations. You can record for many hours. So this is a time series of the avalanche sizes. So I'm parsing my data in words to use the language of previous talk, right? So my words are avalanche. So this is the size of the avalanches in log scale. So let me Fourier transform this thing. Or let me do some deep-trend fluctuation analysis on this signal. What you get is something which looks quite with 1/f spectrum. So this is the Fourier power spectrum as a function of frequency. This is exponent 1. And if you do the trend fluctuation analysis, oh boy, the trend fluctuation analysis, crash course on the trend fluctuation analysis, you bin your data, and for each data you fit a line, and you calculate the residue, so the distance to the line, and you see how this deviation from the line increase with window side, okay? And there's a whole theory to show you, I'm not going to this, that if this goes with the power 1, and then this also goes with the power 1. So this is a way of measuring long-range correlations. This is also a way of measuring long-range correlations. Essentially, if your spectra goes with, if your time correlations goes with tau to the minus gamma, then your spectra go to 1/f to the power alpha, and the relation between gamma and alpha is this one. And if gamma is 1 or close to 1, then gamma is close to 0. If gamma is close to 0, your correlations are infinitely long range in time, which is a signature of a critical process, a critical system. You see, you have these long range correlations in time. Okay, so that looks more like a critical system that I can accept. Now, even nicer than that, as John was saying, you expect scaling in a critical system. So let's look at scaling. I'm going to look at the waiting time distribution. What I'm going to do is the following. Okay, suppose I ask you the following question. Okay, how much I have to wait before another avalanche comes? Answer, well, it depends on the size. Avalanche of size 1, you have all the time. Avalanches of size 100, you have to wait longer. So for a given threshold, SC, this is a parameter here, what is the probability distribution that I have to wait some time tau? Is that clear? Yeah? Okay, so you can calculate this. So this is the density of, probability density of waiting time t, given that the avalanche is size larger than SC. Here is the result. So size 1, size 2, size 3, up to size 36. You see that the larger your threshold for an avalanche, the longer you have to wait on average. It makes sense, right? That the larger threshold, the longer you have to wait. Right? Now, what's interesting is that if you take each of these curves, divided the horizontal axis by the mean time here, so it's tau divided by the mean time of that curve, and multiply that mean time of that curve by the vertical axis. It's just an operation. It's just renormalizing your axis. This is what you get. All the curves fall into the same curve, a unique curve. So what this means is that if I can write this as a function of this is what is written here, and this is the scaling relation that John was telling you about before. We have scaling in the waiting time distributions of in vivo, freely behaving animals, which I think is a nice sign of one possible sign of criticality. In fact, you got even more than that. Since this is dimensionless now, it is dimensionless, okay? And this is also dimensionless. I can compare different rats, different brain regions, different everything. And so before I do this, this is animal 5 awake in the hippocampus during exposure to the animals, to the objects. This is animal 4, slow wave sleep, visual cortex before the exposure. This is animal 1, REM sleep, S1, post-extreme. It doesn't matter. It always collapses. If you anesthetize the animal, then it doesn't. So anesthesia is not sleep. We know that, but right? It is not. It's very different. It's a very different beast. And now, like I promised, since everything here is dimensionless, I can try to see if they all collapse into the single curve. And it does. Well, it's a little thick, but it's biology, okay? And it's so this is where anesthesia doesn't. Anesthesia doesn't. And when you put all the data together, you can see with some goodwill a double power law, which is exactly what you see in earthquakes. You also see a double power law in earthquake distribution of recurrence. Okay, so here I get more decades. Okay, I have like 6 decades here and 7 rats. So it's nice. And obviously this is the exponents for, and the exponents look like the earthquake exponents, okay, these ones. And you can do, this is for all the data, but you can do separately for each individual scenario. And you have slightly different exponents, and you can compare that with the shuffle data, which is the black curves here. And if I put exponents alpha and beta here, the data cluster in one part of parameter space, in the exponent space, whereas shuffle data is everywhere. So this is the best I can call, the best I can come to what I would call universality. So the exponents look the same. Okay? Right. So why did I include this part of the talk in the fall? It's nice result. Yeah, it's a very nice result, but it's not the model. I have no idea how to model that. No idea. And I will bet a dozen beers if the simple models that we've been using can do these kind of things. You have to put lots of ingredients, which I don't know what they are. I bet that you cannot reproduce these results with that kind of model. You take the bet?
During the 20 years that we have been betting beers,
yes.
we have never paid the beers.
But now we have witnesses. It was all in your office before, but now we have witnesses. Okay, it's being recorded, right? So I don't know of any simple model which can do this or this or that. I don't know. Yeah. Log normal. There are several. Well, there are several. One of them is the problem of undersampling. The problem of undersampling, which is what we use to expand the data, right? So it could be that. It could be that. But I'm not sure, because, see, what I'm showing here is a nice result, in the sense that, okay, I modified the model, I included one additional biological experimental constraint, which is undersampling, and I reconcile the model with the data. Looks fine, right? What I'm going to show now, so wait, okay? So I don't know how to answer a question. I don't know how to interpret this. I knew how to interpret this in light of the model, but now I'm going to show that this model has problems. Now here's the end of the fall on the rise on the floor, so to speak, right? So let me revisit the undersampling issue. What I showed you is that this thing here works, so to speak. So if I sample everything, I get beautiful power laws. If I sample only the red, I get this log normal, which looks at the data. Fine, great, wonderful, spectacular. Now, so undersampling changes avalanche statistics, right? That's what we're seeing. This is what Viola Prisman has been saying in a couple of papers. 3 papers here, we also said in this one. Now, why is that so? It's very easy to understand why. Because suppose you have this, I'm sorry for the quality of the figures. Suppose your system is the black, it's all the points here. All the points. It's 1, 2, 3, 4, 6, 7, 8. So 8 by 8, 64 neurons. And your electrodes are the unfilled, are the white dots here, okay? So you have 9 electrodes and 64 neurons. So I create one avalanche here, one time step. So what is the avalanche size that I'm measuring here as the avalanche goes on, okay? So in the full sampling, Okay, I'm measuring 1. This one spiked. Okay? In the full sampling, I measure 1. In the undersample, which is only those 9 here, I measure 0. Nothing happened yet. The spike is here. Okay? So 1, 0. I'm already missing 1 spike. Now, the avalanche goes on to those 2 guys. So this guy spikes, this guy spikes. I see 3 spikes, 1, 2, 3 in the full sampling, and then 1 spike finally in the matrix. So 3 occurred, I measure 1. Now this thing propagates, like this. Now something happened. Look, what happens is that activity propagates here and here, and for the matrix, the avalanche is over. The avalanche is over. While the real avalanche still propagates. So we have more neurons. And it goes on and on and on and on. You can believe me that at the end of the day, in the full system, you have 1 avalanche of size 12. Whereas in the matrix, what you measure is 3 avalanches of sizes 1, 2, and 1. So you see, you lose the tail, and it comes to the other part of the distribution. Just because you're sampling poorly. Now, so undersampling solves one problem, which is reconciling, bending that curve down to the data. Nice. Now, anesthetized rats, or John's data has shown, well, maybe not John's data, because it has a very large matrix. But if you take a normal matrix, 60 electrodes or something, it has the same undersampling problems. It's the same rats, the same electrodes. And experimentally, you see power law distributions. You see? You do see power law-like distributions. This is not a log normal. So experimentally, you see power laws, and it is undersampled. It is undersampled. So how, now I'm desperate. Now the model, the idea we had also, that's so nice. We came up with undersampling, so we bend the curve to what we wanted. Okay, so now bend it back. How do you do that? Can you do that? Can you still have undersampled model and still have this power law distribution? Because that's what we see. For the physicists knowing the statistical mechanics field, note that this is not, I'm not going to do finite size scaling. This is a completely different thing. The system is always huge. What changes is the size of the matrix where I'm measuring things. So, so can I still get power law distributions? Again, power law is not everything. We know that. But the thing is, we do have power laws experimentally. And this has been the model, what I call the workhorse in the field, the theoretical workhorse. So this is how we understand, how we mimic to explain what's going on. So just to be sure, we take 3 different topologies. So this is 2-dimensional in the sense that you have some local connections, some randomness here, but 2-dimensional. 2-dimensional plus some long-distance connections, is small world, and totally random, okay? 3 different topologies. And we're going to make them critical, supercritical, supercritical. Each of those 3. I mean, this is very simple. The brain is more complicated than that, obviously. But can I get something like this? Let's see what happens. So first, you determine the critical point for each of them by making sure the response function is nonlinear and everything works because we can tune the probabilities, okay? This is just to convince you. And then when you sample the whole system, you get beautiful power laws, blah, blah, blah. Just testing. Okay, we can get critical to the networks, critical small world networks, critical random networks. That's fine. Now, let's put in the electrodes. This doesn't count. This is cheating. We are cheating here, theoretically, cheating. Because you don't have access to all your millions of neurons. Let's put electrodes. So right here, I'm fixing the spacing between the electrodes. This would correspond to about 250 micron separation among the electrodes, which is like 8 cell bodies, more or less, which is what we had in those measurements. And you can change, so Lm is the size of the matrix, right? So Lm equals 2, Lm equals 4. Okay? And then you change this thing, and you sample from those things. So what I'm going to show you here is a sequence where you see 2D graphs, small world, random graphs. Subcritical, critical, supercritical. So we're trying everywhere, right? And the different curves are different matrix sizes, right? Electrode matrices. So see, here, 30 by 30, that's 900. How much do you have, John? 500, right? So this is like state of the art. We don't have that. But we tried, in the computer I can do it, right? What we do have is 8 by 8, like the first page. 64. Is it more like a typical number in the literature in the last 10 years? I don't know, right? And essentially, you don't have to understand all the curves. You just have to tell me if you find a single power law here, which is not black. Black is full sampling. Okay? Full sampling is critical here, because I made it critical, of course. But as soon as you undersample it, even if it's super critical, and you undersample it, I know, it's gone. Well, maybe it's not the number of electrodes that matter, maybe it's the spacing. So, okay, let's keep the number of electrodes fixed and change the spacing. Okay, I can do that, right, in the computer. No power law, no power law, no power law, no power law, no power law. If there is one we couldn't find. This is a power law? This one? Nah, but this isn't. Yeah, but this exponent will be like -7 or something. I don't know. The exponent should be the same, right? We get 3/2 exponent experimentally, right? So, and we can do a last resort, which, okay, maybe you have to rescale your time. That's not how you do with the data. The data well, let's do like this. Let's choose an optimal being determined by the data and try the best configuration that we find. Or maybe the configuration that we use in the experiments, which is this one, which is like 4 by 4 matrices. And you see, if the system is subcritical, okay, so this is the data, right? The black curve is the data. The red curve is the best critical model that which we could get, which looks okay, but it's very bad, actually, because it doesn't have the cutoff. The cutoff is very important for scaling purposes. And the data doesn't have that. This model, I'm sorry, doesn't have that. The model doesn't have that. And supercritical, it's kind of crappy. If you try to fit this function with what we did to fit the experimental data with like a power law with an exponential cutoff and some exponent in the cutoff, if you fit this thing and compare what you get for the data, for the parameter tau and the cutoff here, they are somewhere like here, whereas models are far away. It just doesn't work, which is kind of strange because it's a very simple test of a model if you want to compare with the data. The data is not fully sampled, right? So, are critical branching processes or critical or dielectric percolation kind of model satisfactory models of neuronal avalanches? Well, only if fully sampled. And even like this, well, with some limitations. So, what are the pros? Well, if you do the calculations for the fully sampled, you get neuronal avalanche exponents which agree with the data. So 3/2 for size distribution, 2 for duration of the avalanches. The response exponents, like I shown before, 1/2 agrees with psychophysical responses with sensory systems. That's nice. And like I said, it has been a theoretical workhorse in the field over a decade. So it has given us useful picture to talk about. So, you know, it propagates more, it propagates less with my hands, right? But I think that's as much juice as we could extract from this theory, I think. For instance, it fails with undersampling, like I've shown you now. We found no power laws. I know the power law is not everything, but we didn't even find power laws, right? Power laws are supposed to be easy, right? So separation of time scales has to be imposed by hand. And remember, this keeps bothering me. And I'll come back, maybe. there are no time correlations between consecutive avalanches in this kind of model. And we see that experimentally in free behaving animals you do have. So there are different perspectives. So maybe if you use a different topology, if you scale the connectivity with n to 1/2 instead of order 1, these are technicalities. Some people have shown, like Miguel Munoz, that if you have hierarchical networks, so a very particular topology, you can have something which is called a Griffith's phase, which is not exactly a critical point, but which have generic power laws. Then maybe what we're seeing is not a critical point, but a Griffith's phase. I don't know. And there's also a very nice paper by the group of Link and Carl-Hansen in Amsterdam, where they show not excitable neurons, as I've shown you, but oscillators that eventually, if you tune the parameters, will undergo collective oscillations. Then right at the transition where these collective oscillations emerge, you can have this almost oscillation going up and down, then again going up and down, and if you put some threshold and call this going up and down an avalanche, and do the statistics for a very small system, you get the 3/2 exponent. So if this is correct, if I can scale up this model, and I'm working right now with a student doing this, that will mean that the phase transition is not one from silence to non-silence. It's something else. Something quiet to oscillations, like we see a glimpse of in those slices, those oscillations I've shown you. So there's room for theory here. See, we need models that can give a phase transition, and we don't know what the phase transition is. We thought we did, or I thought I did, and I think we were wrong. I was wrong. Okay? And I think I forgot to talk about Don Katz's comments on the modularity. I forgot, right? So the thing is, why this criticality hypothesis, what does it have to do with what you were talking about before, is that at least in the hand wave space, you could possibly conceive that this critical state having these long-range correlations in space and time and creating those islands that John showed us on top of an anatomically modular structure could solve for the modularity issues. But dynamically, you don't put it, not everything is in the anatomy, but in the dynamical regime, which creates those modules. It's a functional modularity, exactly, which is very diverse. Actually, it's the place where you have the largest diversity of metastable state. It will give you flexibility and potentially solve this integration versus segregation problem. Well, visual cortex is here, auditory cortex is there, but I see the guy singing, and I know it's the guy singing, right? The same guy. That I see here and listen here, it's the same guy. So these long-range correlations, I don't know, could be an alternative to synchronous firing, which has other kinds of problems. That's the link that I'm sorry, I should have done it before, but okay. Okay, thank you very much. Just some advertisement. We have a new electrophysiology lab in Recife. This is real experiments, real students, real data, real electrodes. We need students and postdocs. And also some other advertisement. This year, I don't know when, November, there will be an electrophysiology course in Recife. So if you're interested in coming, please look at this site here. They're also on Facebook and enroll. I'll take your questions if there's any.
Okay, so we have time for questions before going for lunch.
Okay. Okay.
So just getting back to your directed percolation model. In real directed percolation, the actual parameter which gets you towards criticality or away from it is the concentration. Is that something you've taken across to say it has something to do with the connectivity of neurons?
Say that again.
So in real directed percolation, the main parameter which gets you on either side of criticality is the concentration of connected sites, right? On some backbone or something, right?
Yeah.
So in the case of the neuronal network, is it to do with real connections or something?
No. But you talk about you're talking about percolation. In directed percolation, it is the connectivity, right? The probability that you connect once you are there. So that's the difference between percolation and direct percolation, right? Well, yeah, I mean, given a backbone topology, the probability that you connect, given a topology, will change, will give you a different level, right? But here it's just interpreted as the way, as the effective connectivity between neurons. So, say the probability that I transmit a spike, taking into account everything, all my ignorance about excitation and inhibition, which is not in the model in detail. I'm not sure I answered your question. Did I? More or less? Yeah.
Mauro. Yeah. About the undersampling.
Yes.
It's very difficult to wait for the Italians until they have more electrodes. So what I suggest is that you look at the data and still undersample it even further and see how the curves change as you just consider a few electrodes and then more, and then you go up to the full number of electrodes that you have and see if you can have a scaling derived from that and take then the limit to infinite number of electrodes.
It's a very nice idea. We tried it actually, but we didn't. You see, finding a scaling, it's always a trouble because you have to get it from somewhere, right? So we tried a couple of obvious choices for scaling. We didn't find any that would collapse the data. On top of that, we have very few electrodes because you see, these are old experiments by Siddhartha, so they were implanting V1, S1, hippocampus. So if you look at, say, V1, of all the 36 electrodes they could have, only 20 are in. So we don't have that many electrodes. So the scaling is limited by that constraint. But even within that constraint, we tried it, something like what you're saying. We didn't find scaling in the data. I don't know, which is very troubling.
No, but in the simulations, you can do that.
Oh, you mean undersample even further, you mean?
Yeah.
Here. Yes, but
so you did that. Then
yeah, we went up to 2 by 2.
2 by 2.
This is 2 by 2, right? And
And
you mean try to rescale those distributions. We couldn't do it. I mean, it doesn't scale well. I mean, we didn't find a scale that worked. Yeah. It looks like you should be able to, right? Just couldn't find it.
One real quick question. So the data that you got from the anesthetized animal, you showed that it didn't have a shape collapse for the waiting times? So it was not critical, is that correct? So when it's anesthetized, it's not critical?
Well, excellent question. Since I know who the referees of the paper were, I know it wasn't you, but one of the questions of the referees.
I was for an earlier version, and I was actually favorable of it. And then it got sent someplace else. But I'm just trying to understand what you're saying.
There's no easy answer for this question. Essentially, I can resume like this. So if I look at, if I look at this time, let's call it time domain data, okay?
Sure, yeah.
Time domain results. The freely behaving animals have signatures of criticality. Scaling, nice scaling. Also this 1/f spectra.
Right.
Whereas anesthesia, I didn't show you, but it doesn't have 1/f spectra.
Right.
It's like a Poisson process, like 1/f squared, right?
Right.
And if you do DFA, this is not even a straight line.
Right.
We just put in a straight line so that we can show that the data is not a straight line.
Sure.
And if it were, it would have a crappy exponent, right?
I agree.
Let's say in time domain, I don't see any signs of criticality in anesthesia, whatever that means,
right.
what I'm saying. And if I forget time domain, and I look at the classical criticality signature, historically classical, which is size distribution, then it's the opposite. In vivo freely behaving animals do not have power law size distributions, where anesthesia,
They do.
they do.
Right.
So the consistency that you are talking about, we don't have it.
Right. So I just want to make sure I understand where you're coming from. So you're saying that the data support the idea of criticality when the animal is awake and behaving
In the time domain,
in the time domain, but the branching model can't account for all this stuff.
yes.
Yeah, I agree with you. Yeah.
Yeah.
Okay, I just wanted to make sure I
Yeah.
totally understood.
But not only that,
yeah.
the branching model cannot account for the power law, for the simple power law size distributions, as soon as you undersample the model. This is undersample data. Do you agree?
Yeah.
This is spikes, and this is 20 by 20 or 30 by 30 electrode
Right.
matrix, right? So this is severely undersampled, and I see a power law
Right.
experimentally.
But that animal is under anesthesia, which you
Yes.
said was not critical. Is it super critical?
I don't know. It looks like a power law to me. Well,
No,
more or less.
But based on your time
Yeah.
collapse,
But
you
based
said
on-
that it wasn't critical. So I guess what
No,
I'm
no,
wondering,
I-
is it supercritical or subcritical?
No, I have no idea. Look,
Okay.
see, is the in vivo free behaving animal critical? I don't know. I see signatures in the time domain, but I don't see other signatures in the size distribution. In anesthesia, I see this signature. It's like this size distribution, but I don't see anything in time domain, like they were uncorrelated one to the other, which sounds reasonable for anesthesia, right? Maybe they should be correlated. What I mean is that, see, this is the basic stuff that we always started with, power law size distributions. And I always thought we had at least a model for that, which is this, okay, this bunch of neurons which spike and you can control and it's critical. And what I'm saying here that, well, put another ingredient and you lose even that.
Sure. Well, let's talk during lunch then, okay?
Yeah.
Thank you.
Thank you. Is it over?